Thanks for joining. Welcome to tonight's Michigan State University Beekeeping Office Hours webinar. This is our April 2026 webinar, and I'm here with Dr. Zachary Wong, Dr. Keith Mason, Dr. Megan Milbrath, and Dan Wines. So this program is part of MSU Extension, and our programs are open to everyone. So I'm going to start us off with some announcements. Then Keith Mason is going to give us a weather update. This is a new segment that we're really excited for this year. We just received a grant from Project GREEEN and are going to be providing more weather information to beekeepers. And then our team will share some beekeeping updates about what we're doing in our bees, and then we will respond to your questions. So you can find a lot of information and resources on the pollinators.msu.edu website. Same thing with the canr.msu.edu/bees website. And if tonight's webinar is being recorded, and we'll post it to the MSU Extension website and to our YouTube channel. So that's the Michigan State University Beekeeping YouTube channel. A couple of past webinar recordings that we've done somewhat recently that we wanted to highlight was that late last year, Megan Milbrath and Peter Fowler did a webinar on European foul brood. Many of you are aware that there's a lot of great research happening here on European foul brood, and they shared updates and a lot of things that they've learned about that disease. And then in February, Megan Milbrath and Zachary Wong did a research update. So they talked about some of the research projects that they're doing, and that's a recorded webinar that you can find on our YouTube channel and website. We have monthly resource articles from April through September that share some of the resources that are relevant for beekeepers based on the time of year. So here is the link for the April resources. And then our National Beekeeping Survey is open through this month of April. So this is a survey that happens every year and asks beekeepers about their colony losses and management. So we hope that we get many responses from Michigan to help us understand how bees are doing. All right, and then we at Michigan State are really excited to be hosting the Heartland Apicultural Society Conference Saturday, May 30th and Sunday, May 31st. That'll be on campus. There is a lot we have planned. There are social events. We're going to do a tour of our facility the Friday before the conference. There is going to be 3 different apiaries on site near the conference center where we'll do hands-on activities. Some of the classes you need to register for ahead of time, and I will say that if you want the best selection of options, it's best to register early. Some of the classes are already over half full, so hope people sign up early if you want to have those as options. There's going to be some laboratory workshops. Zach is doing a lab, as well as Peter Fowler. And we have 4 different plenary speakers from out of state. We have breakout sessions. A lot is planned for this. We're really excited about it. So we're hoping to see many of you there, and registration is open. Another conference that I'm pretty excited about is a new one that we're doing with our consumer horticulture educators. So those are the educators that work a lot with home gardeners, and it's called Healthy Habitats and Productive Pollinators. So this is a webinar that's about, this is a conference that's all about our wild species of bees, the species that, aren't honeybees, but are just part of our ecosystems and really important. The keynote presenter for this conference is Dr. Elena Evans from University of Minnesota. She's a fantastic presenter who's done a ton of really cool research with bumblebees and definitely an expert in her field. So this is an in-person conference in Flint, and registration's open for that. It's a one-day conference. All right, and we want to acknowledge our funding that supports this program from Michigan Department of Agriculture and Rural Development, from USDA NEFA, from Project GREEEN. So happy to have that support. And if you have questions during the webinar, you can put them in the Q&A box of the Zoom webinar. If you have questions after tonight's webinar, you can submit them to us through the form at the Ask Extension form at pollinators.msu.edu/questions. That's really the best way for our team to receive beekeeping questions outside of the webinars. All right, so I'm going to turn it over now to Keith.
Hey, thank you. I'm gonna pull up some slides here. Yeah, let's do there. There we go. Right, so let's see if we got that right. Yeah, I'm sharing the big screen. Okay, good. Well,
Great.
thanks everybody, and thanks for inviting me. And I'm really excited to be part of this. I usually come at things from the crop side. I'm actually an entomologist, but I'm the coordinator of the EnviroWeather program. So there's a lot of weather data that's going to be really important for beekeepers as well. What I want to do is to get information from you guys about what types of things you need is part of what I'm trying to do here because we do have a new project as Anna mentioned. Yeah, so it is a Project Green. You don't have to read all this stuff. I'll give you a general thing. But yeah, we put together, we know that there are tools that we could probably put together fairly simply with using our crop bloom dates and then also other published research about when nectar plants are blooming. So we want to try to integrate those kinds of things with a little bit of forecast as well, since everything, everybody's hinging on the forecast. So that's one of the things that I want to try to bring into these. But if you have ideas, let me know, or put it in the chat or what have you. Um, but yeah, but so part of that is for me to join these meetings here, and I'll do that regularly and try to provide good weather for you. Well, good weather data. Who knows if it's going to be good weather? But and then as we develop the bloom models here with this project, we'll get feedback from you beekeepers and from the team as well. So we're looking to have good interactions with all this. Okay, so just to kind of start with where we were at. So this is the average temperatures over the last week, ending yesterday, and there's a pretty striped pattern here in Michigan where you've got obviously warmer temperatures. So we were in the 50s according to the scale here as an average, and then it gets cooler as you go up, and there even were still some 20s and teens in these in the upper UP. Um, so that's just, looking at what the temperatures were. We also like to think of things as what's the temperature now, kind of in reference to what an average temperature would be, or that typical Michigan spring is what I've been chasing for the 20 years I've been here. But I don't know that that exists. But this lets us look at this in reference to other years. So again, you see a lot of the same type of pattern. So this is the temperature departure from normal. And so these are uh, showing actually in Fahrenheit degrees here. So these darker areas were almost 20 degrees higher as an average temperature over this week span here. And yeah, much cooler up north. So there's definitely a division in the state now where things up north are cooler than in the south, as we would imagine. But we track that with growing degree days. And on the left here is just the accumulated growing degree days. This is a simple base 50. And you can see that, the differences were almost at 130 down in the lower tier and, single digits up in the UP. And again, so these are the accumulated growing degree days. And then this is growing degree days compared to where we were in years past. So you can see that we're almost 14 days ahead in the south and almost 14 days behind in the north. So again, we have this big dichotomy of above and or ahead and behind in growing degree days. So depending on where you are, that can tell you what your year is like. This is looking at the precipitation over the last week as well. And there was quite a bit of it. This is actually the measurements of the actual precipitation that fell, and a lot of areas had 2.5 or 3 inches of rain this past week. So we've definitely recharged our soils with a lot of this. And then if we look at that according to how much rain we normally get at this time of year, we're in the 1.5 to 4 inch difference overall. And this is really important because like March or April, we only get 3 or 4 inches total during the month. And so now here we've got, in this first week, we've already got the rainfall we would normally get in March or April. So we're going to be very wet for a little bit here. And again, this is our rainfall forecast. So this is the total amount we expect to fall in the next 7 days or so. And here we have a wide range against across the state as well. We're going to have some in the southeast, some lower totals, some maybe less than a tenth of an inch, compared to up to 2 inches up in some of the northern areas here. And now when we also want to look farther out too, or at least that was kind of part of the information that I got initially. And we usually do those in what are called these outlooks. They don't tell you the exact temperature that it's going to be, but again, it works on this normal thing. So the temperature outlook on the left-hand side over the next 6 to 10 days, we're going to be warmer than normal. So it'll be kind of warmer than it normally is in April. And if we look on the right side, we're also going to be wetter than normal. So warmer and wetter is kind of what we're looking at. We've got the same kind of outlook for 8 to 14 days as well. So I'm not going to show you that one, but I'll show you the month of April, which has a little bit of a change. So here the last month, or the last look we had, it was saying we were going to be warmer than normal. Now, if we look at the whole month averaged out, that can be we don't really have a strong signal for it being warmer or cooler than normal. So we're just in the normal range for heat. But for precipitation, we're going to be still a little wetter than normal in that over this period here, over the next month. And they do a seasonal look at this as well, which does change, so we look at these each month and see how things have changed, but that kind of equal chances of normal temperatures are there. And we're starting to see that in precipitation as well, although most of Michigan is still going to be wetter than normal, is according to the models at this point. So yeah, just a real quick summary. Obviously, I didn't have much stuff here, but warmer and wetter is what we're looking at. That's probably going to continue through the end of the month, and then if you look growing degree days, we're 14 days behind up north, 14 days ahead in the south. So go to the south. No, and so this also is my time to uh, if you guys are interested in other sorts of forecasts, other pieces of weather. So, I know we've talked about wind because bees don't fly in the wind. You've got, I mean, soil temperature, soil moisture has a lot to do with plants and their emergence. So, I can bring those kinds of things in as well. So yeah, if you're looking for something in particular, let me know because we can definitely gear it more towards beekeepers here. And I had a bunch of weather links if you guys are weather geeks, which, I am, even though I'm an entomologist too. But yeah, if you are, this synoptic data weather map is a really cool thing to check out because it has it actually has the entire world, over 100 networks of weather. So, and you can go to it, it's free to use. And I could do a segment on that if you guys are interested there. But yeah, that's really all that I've got. So I'm just going to kind of hang out for a little bit to see if there are some questions.
So, and I did just put Keith's links in the chat, including the synoptic data weather map. And then there is a question that when you were talking about the degree days that says, how do you decide if you're going to post the growing degree days that are calculated for 42 or 50 or other? And maybe if you wanted to maybe just give a super brief overview of, I'm not sure that everyone on the call is going to be familiar with growing degree days.
Sure, sure, great. So yeah, growing degree days are really just a way to measure the amount of heat over time. So we want to know how hot it is for how long because that heat accumulates or heat accumulation drives insect development and it also drives plant development. So we can use growing degree days to estimate when things are going to happen in the field. Um, Honeybees, they don't really go by growing degree days because they can warm their own hives, right? So they're not as dependent on temperature as other insects are. But still, yeah, it's a way to go, to count how much heat you had over a period of time. And where those bases come in, the 50 versus 42 or 45, it really relates back to the research that was done to develop the models. So the base temperature is the temperature below which no development happens, but above is where things start to happen. And that's just straight biology because we've got all the metabolic processes are dependent on temperature. So this is, more heat, things are going to happen faster. So things grow faster, the proteins are working faster, all that stuff. So that really depends um, on the research. And 50 degrees is just a really commonly used one for a lot of fruit crops, I think, and actual vegetable crops too. But yes, it's really when temperatures are above that critical threshold. Those are the degrees that count. So that's why we use growing degree days.
And
Hope that
there,
helped.
no, that was fantastic. And there was a comment, I think, when you were asking for things that beekeepers are interested in, and someone put the growing degrees for the major nectar flows, which is great
Okay.
because that's what we wrote the grant about. So
Okay. Yeah,
um,
no, absolutely. Yeah, I guess yeah, we're going to look at the plants that we have, the nectar plants, and see what their bases are. And then, there's published research on a lot of things. And I was really glad to hear that say that you're involved with the consumer hort people because they're going to have a lot of this information. So that's wonderful that we've got those connections too. So great.
and also, if people do have weather questions, Keith may not stick around the whole night, so you can get him in now or every month when he joins us.
Right.
There is also a question that is wondering if you have a good site for pollen counts or availability. They say they know they have some for folks who have allergies. So I don't know if you want to say.
Hmm. Oh, yeah, I know there are some of those counts. We have never worked with them, but I can certainly look into it to see what if things are available that we could use.
And then there's just one more comment where someone really, they said it was a nice presentation, but they really were hoping that you would learn how to control the weather, which I appreciate.
Thank you. That is actually, yeah, that's one grant I can't get funded. But yes, thank you for that. That's good. That made my night.
Yeah.
And I put my email in the chat too. So if you have further questions, you can go ahead and oh, I just sent that to hosts and panelists. Actually,
Yeah, I'm fixing it for you. I got
okay. Thank you.
So Keith, I have a question. So
Yeah.
in near the campus area, were the fruit trees bloom earlier this year than usual? As
Yeah, well, if we keep going with the growing degree days like we are now, yeah, we should see things a week to 2 weeks early. That could certainly advance that.
my 19 cherries are budding, the flower buds are
You can
almost
see
coming
flower
on.
buds already?
Yeah, I
Okay,
did.
yeah. Yeah,
And
things are definitely happening.
I need to check my pictures from last year, see when did they bloom. See if they're early entry this year. Because the weather is so weird. One day it's getting
Right?
70s, like 3 weeks ago. One day it was 70s and bees were bringing pollen, and now it's like it's snowing today, and
I know
they
it's
almost
crazy. Just wait a minute. You'll get the
yeah,
weather you want.
yeah. And then one year I had my Asian pears bloom. I think it's 2 years ago, blooming early, and then there was a cold, like killed all the flowers. Yeah. Yeah. So yeah, going too
Right.
early
Yeah,
is
that's what
it's
happens.
bad. Going too early is bad for the fruit trees, I think. Yeah. Yeah.
Thanks, Keith. That was great.
Yeah.
Really appreciate it. All right, so now maybe we can just do a quick update on what we're seeing or what we're doing in hives right now. Dan, do you want to start us off?
Yeah, so we're in early April, and that to me it's really especially with the volatile weather, kind of a patience time of year. It's all going to be a million miles an hour here in a matter of a couple weeks. Um, but right now, the main things I've been doing the last few weeks is feeding bees, not looking to stimulate, but preventing starvation. With this volatile weather, they are getting some flying time in, seeing pollen coming in. But had bees, colonies that came through the winter light, they got dry sugar in February and early March. And then as we got into later March, I've transitioned to liquid feed as needed to kind of keep them going until, dandelions and some other things if we get nectar coming in from that. So a lot of the tasks, cleaning bottom boards and that, but I'm not really manipulating brood or anything. I feel like that's where the patience comes in of I'm going to do more harm than good. So it's really just keeping them well fed and looking probably at splitting in 3 weeks, usually right around the end of April into early May, depending on how the timing of things go the next few weeks. But that's kind of what I've got lined up.
Hey, Megan or Zach, do either of you want to chime in with what you're seeing or what you're doing?
Megan?
Well, I went
So,
directly from the UP down to Georgia and haven't made it back up yet. So I'll tell you what I'm planning on doing when I get back is this is, and I will tell you what I'm telling people too. So this is the time of year that I always joke that I'm just going to turn my voicemail to just say stop digging in there because I'm already getting lots of requests for queens from people that feel like their colony might be broodless or think that it is. I don't go down into the brood nest at this time of year because we do continue to get cold and freezing, and I don't want to just I the more I can leave them alone to just start raising brood, the better. The one thing that I will do is go out and put boxes of drawn comb on all of my big hives. And so I have gone out and looked to make sure everybody has food, and mine are all sitting pretty heavy, thankfully. And I have taken out the few dead outs and redistributed the honey from that. So I'm not feeding, which means that I can put drawn comb on top. And that does a few things that allows the bees to clean it up and get it ready to go. But it means that if there is nectar coming in, like when we do get those warm days, they put it above the brood nest. And then I have it already out there to go when I'm doing the splits. So like Dan said, we're a few weeks out from doing splits. And so until I'm actually going to be looking to see if they're ready to be split, which I know they're not now, I'm just not going to be digging in the brood nest. So, but I have already given out that message multiple times. If I had gone in there and saw that it was completely queenless, I would just stack it on another hive and move on. There is a question for Dan, and it asks, when you're doing your splitting, are you going to do with purchase queens or are you going to let the splits requeen?
Yeah, so most of that's going to be purchased queens. Certainly anything that I'm doing around, end of April into the, early May. those will be purchased queens. If we do some splits as we get into later May, early June, depending on numbers, if I need more, I'm a little more comfortable splitting then with either a cell or letting them raise their own with, drone availability and mating weather in Michigan. But I don't want to try to get a queen mated locally, given our, volatile early May weather and say if they don't have the drones ready. So purchase queens.
And then someone says, where do you get your queens and what type?
I have some coming from California, some coming from Texas. Uh, some we're gonna get some Italian bees this year. I have one project that I want bees that lay later into the season are a little maybe less responsive to the environmental changes. So I'm gonna run at least a yard of Italian bees because we've had some of the darker carniolan bees and still like them. Big fan. I just have one particular project where I want bees to ideally they would brood all through the winter. I know that's not going to happen, but we want to extend that season as long as possible. And Italians kind of have that reputation for being, less responsive when the food shuts off. So as a beekeeper, you got to provide more food. They tend to winter a little bigger, more brood, but it means they eat more. So pros and cons, but we're going to go with some of those this year. But having said that, we've got a variety of genetics. I'm not really working towards any one bigger genetic goal. It's kind of depending on what we've got going on, and I like to get from a number of sources just to kind of keep things mixed up a little bit, little variety, and given we're not running a breeding program internally
Yet.
yet.
Um, Zachary, did you have anything to add about spring management? There are some more questions coming in.
So I'm not really working with bees now since Dan has taken care of our bees. So I've been writing papers mostly last 2 or 3 months.
And some
Besides
very fun papers too.
thank you. And I did do one small experiment in Mississippi because in January it was too cold in Michigan. And we did have that warm day. I was trying to do some experiment, but the bees are not totally normal compared to summer bees. They're coming back, and I have something in front of the beehive. They don't want to come and land. It's like something's different, and then they're just hurrying. So I didn't get data. Yeah. That was 2 weeks ago. Yeah. It was a warm day. Yeah. So I guess I have to wait for another month before we could do flight experiments if we need to. Yeah.
So there is a question about when should I put Viroxan on in 2 weeks, question mark. And that's actually, so I'll deal with that first, and then if someone else I can see Anna wants to do, because I usually give her all the Varroa questions, but I'm actually down in Georgia working on a trial. So last year we did a trial with some California/North Dakota bees looking at lots of different treatments. And we looked at like a set of treatments that were put on in May, a set of treatments that were put on in June, and a set of treatments that were put on in August. And one of the things we found were that even by May, the mites were already pretty high. And these are commercial colonies that are out in almonds, so they're not really shutting down. So it makes sense that they're higher compared to the ones, that would be stationary in Michigan. And so this year we are doing treatments in early spring associated with splits. So we had half the team out in California and putting mite treatments on when people are making up splits with queen cells, and then we're doing the same thing down here. So I'm telling you that what we're looking into, that we don't have data for it obviously yet. So what we're trying is at the time that you're making splits with queen cells, so you're also going to get a break in the brood cycle, we put in Viroxan the same day we added the queen cell. And that's one of the options you can do. We were making up about 4 frame splits. And so you generally put in 1 strip of Viroxan per every 2.5 frames of bees, so it's per the label. And it says that you can leave that amount in as the colony grows. So we put 2 strips in generally. The other treatments we're trying are Naroa, an oxalic acid dribble, and Apivar 2.0. So I haven't really messed around with putting Viroxan in the brood nest at the time we're making splits. One of the nice things it is a 56-day treatment. You can leave it in there. One of the things you do have to be careful is that the bees do tend to not raise brood, or there's kind of an issue with directly behind the strip. So we had to be really careful. Like if you've got an itty bitty brood nest, you don't want to put those strips right in the middle of the brood nest because that could really hamper early spring growth. So it's a thing that we are trying this year. So I don't know about using it early because I haven't done it yet. Where I did use it last year and was pretty happy with was putting it on the same time as Formic Pro at when I put my honey supers on. So a little later and then counting on that late treatment through the summertime when my honey supers are on. Anna, did you want to add to that or anyone else?
That sounds good.
All right, I'll give you the next one, Anna. So Mike says, I overwintered with double deeps. If my bees, which are mostly 5 to 6 frames, are all in the top box, should I reduce to a single deep?
So I would say no, they're going to be needing more space really soon. Sometimes beekeepers do like to do a reversal. If there's no brood in the bottom box, you can just take that bottom box and put it on top, and then the bees move into it and use it for brood rearing and storing nectar. You can also just leave it on the bottom and they'll also move down into it too. So that works just fine. But I would say this time of year, we're thinking about our bees are going to need more space. The exception is if you have just a colony that's really small in population. Dan can kind of talk through that maybe a little bit because there's been some colonies that are basically dinks, really, just a couple frames of bees. And in that case, it probably does help to give them a smaller amount of space to manage. But um, for that situation of having bees in both boxes, I would just leave them for now. And Dan, do you want to talk a little bit about what you're doing with some of the really small colonies?
Yeah, so, and this is, completely optional, but as I've kind of made the rounds, starting mid-March, I would say, kind of picking up dead outs and making, assessments on where things were at. I have reduced down some of the really small colonies, and in that I'd say, cantaloupe or smaller, kind of a, 2-ish frames of bees. And I've either left those in a single deep or I've had some really small ones like a grapefruit worth of bees, and I put some of those in 5-frame nuke boxes. Just because it is small. That's the case where, like Anna mentioned a minute ago, like 5 or 6 frames of bees, that's going to take off over the next few weeks and month. That's going to be into that second box before you know it. Something that's under a couple frames of bees, I find that's kind of the level where they just don't ever have that critical mass. They can't maintain a big enough brood nest where to max out the queen's laying ability. Their bee numbers are their limiting factor. They're never going to get bigger without a little bit of help. So that's a case where I do think they can do a little better in a smaller space. Also reducing that entrance down. Not that robbing is a major concern in the spring, but I do like the little, the ones that have kind of just limped through the winter. I do like to give them every little extra advantage I can if I judge it's worth the time to, save them. Some people are the philosophy sink or swim, and that's fine as well. Um, One thing you can also do in a situation like that with really small colonies is if it's queen right and it's just small and you're just limited in the brood nest development based on number of bees to care for that brood and keep it warm, is you can trade positions colonies. You can take one that's, a 6 or 7 frame colony and trade it positions with something that's a frame or two of bees, and that's going to gain a little bit of population where it just helps get it over that hump. And then maybe in a couple weeks the population's grown. Maybe you can give it a frame of emergent brood that's going to hatch out, where you could kind of baby step it along. But again, it's personal decision where how much do you want to invest in these little ones versus spend your time and all your resources in your big productive ones. That's totally fine too. So that's just personal preference and beekeeping philosophy.
So Anna had sent a couple of the recent questions from Ask Extension, and one of them says they're planning on trying to overwinter 6 frame nukes this coming year. What is the best overwinter setup for nukes? I added to the chat a report that I did. So I did a Sayre Farmer Rancher grant basically on that question in 2016, which now I'm realizing is a very long time ago. But I did 2 years and I like tried to overwinter nukes in lots of different configurations. And the main thing that came from that report is that it really just depends on how you want to use the nukes in the spring. So I had pretty good survival in lots of different configurations, and I compared. So I was doing 5 frame nukes to start with, but I did polystyrene nukes, the Michael Palmer method, some custom nukes that I had made that were like 4/4, single deeps, and I think just full size hives was the other comparison. And I didn't see a huge difference in survival. What I really saw a huge difference is how easy they were to use in the spring. So the ones I don't like having the ones that you have to transfer out of equipment more than you have to. It's just a pain to always be moving stuff from equipment. So honestly, I just found if you're going to overwinter 6 frames, having them in a single deep was by far the easiest. Because then for growth of the spring, you just add another box. For transportability, the polystyrene ones were the easiest. But in terms of survival, I would say use the things that you've already purchased are also really good for that. I don't know if I think I probably do the most nuke over. Oh, Anna, you want to say something?
Well, when you were giving those examples, Megan, what was the smallest or lowest number of frames that you overwintered? The smallest.
So the polystyrene ones are 5 over, and I would say the smaller number of frames you go, kind of the fussier they are to manage. Because like if you start those polystyrene ones too early, they'll just like explode. And if you start them too late, then you're kind of dealing with a small amount of bees. They also don't make any of their own food, so you kind of have to like put a full honey frame, or not any, but like you don't count on them on building up. Um, so yeah, I was doing the first year that I did it, I tried to make them all up with kind of uniform 5 frame nuke sizes. There are a lot of people, though, that what they'll do is they'll make up like a 2 frame nuke mid-July and allow it to grow. And then it does get bigger than 6 frames at that at that point. So a lot of times you can, if you put it in something where you're making it small and allowing it to expand. So there was a question if it was a single box or double box. It so I did single deeps that were literally just single deeps, and that actually came of when I went to China and got to visit a bunch of beekeepers who were just overwintering nukes, but they were using single deeps and they just had a follower board. And so what I did is I just overwintered a single deep and I just used sheets of foundation rather than a follower board. And that was lovely because then they could just grow. The polystyrene boxes that were only 5 frames, when they exploded in the summer, you just had to be like really, really watching them closely. Like if you didn't drop them into a full size hive, they just exploded in the summertime. All right, so Anna, there was a question. Um, let's see. Wait, I'm going to read the one that you just put in the chat and give it and have you answer it. So it says, my colonies overwintered with a deep and a medium. All of the bees seem to be at the top in the medium. Classic. How do I get them to move back down to the deep before I try to do a split? I need to feed them and not sure how to configure the colonies to feed them and give them room. Very classic scenario.
Yeah, so I'd say that's normal, right? As colonies consume honey through the winter, they often end up in the top box. And they, as they grow, so when we have nectar coming into the hives and as the the brood nest expands, they will just move back down to and start using that box, that bottom deep box. And I don't feel like this is a problem that needs to be fixed. We're just now thinking about both that deep and that medium as both brood boxes, and you'll wait until the colony is the right size to split and it has the right amount of brood, and you'll just move some of those deep frames into a split if that's how you want to do it. You could also have two hives that each have a deep and a medium, or you could leave the medium on on the original hive or move it to the split. There's a lot of easy ways to do it, but not something that we have to really worry about. You should still be able to feed them just as you normally would.
And one of the resources that I added to the chat is a system for making splits that's really, really easy. And I didn't invent it and I don't know who does, but it's basically you just alternate the frames. So if you have a deep and a medium, as Anna mentioned, one of your options is just to kind of put half the frames into another deep and medium setup so that you have them all together.
I didn't see that come through the chat, Megan. Did you see?
I might have just sent that. Let's see. It is called. I'll put that back in there. Oh, it's called this. It's the Resources for Beekeepers Swarm Management handout, but I'll put it in again because I've been getting kind of wild with the links that are going in. Actually, the next one I'm going to put in first is another question that came in. So I'm posting the Apiary Inspectors of America, the AIA has a lab list. So there was a question that came in about Nosema. And so the first part of the question is whether or not MSU does any treatments or testing for Nosema apis or Nosema ceranae during the spring or the fall. And I've been working since 2017 to get the veterinary diagnostics lab to take samples. We are very close, but we are not taking samples yet from beekeepers. However, the link that I just put in the chat, which is hosted by the AIA, does list labs that do take samples. And then I think, Zachary, since you just published a paper on Nosema, the second part of the question says, additionally, has Nosema been a significant issue for apiaries in Michigan, either historically or currently? Do you want to talk about Nosema, Zachary, and spring in Michigan colonies? And you're muted while you're thinking.
Additionally, like when Roger was here before I was hired, that was when Nosema apis was the main issue. There's a lot of talk about Nosema being a silent killer because you don't see them with your naked eye, and then you see all those application in front of the beehive and very slow spring build up. So then, of course, Nosema ceranae comes, and initially there's a lot of news because the Spanish guys saying they kill the bees right away. And of course, probably the Spanish bees are more sensitive, I think. And now Megan and I, we published 2 papers together. We don't see that quick cure like the Spanish guys like die in a week. So we did see a shift of 4 or 5 days shorter lives Nosema infected bees. So you would think it's small, but actually for a bee during the summer, survival of 35 days, 4 or 5 days is a big deal. That's half of your foraging life. So, but since then, Nosema has been quiet down, and we pay so much attention to raw mites, really. So we sort of lost track. Nobody's really studying to see if you don't treat, do you see a higher mortality? For example, nobody's treating now for the last 5 or 10 years, and we don't seem to see a big effect. Of course, we don't have the control group that will feed fumagillin, first day in spring. We used to feed them prophylactically with packaged bees. And now, of course, do we need do we need the veterinary prescription for Nosema drug? For Nosema drug, it's just been so expensive nowadays since we they were stopped producing for a while, and a Canadian company picked up, and after that the price I think increased 50% or something. so so now I think most people don't treat. So it's basically we don't know if it's making a difference. But I'll say it doesn't make that huge difference. For example, it's not making a 20% difference. Does it make 5%? We don't know. Yeah. So that would be my gut feeling is that could be a small effect helping slow building up in spring if you have when you will have Nosema, they are like ubiquitous. And when I was studying Nosema 20 years ago, we sampled colonies. So we started with brand new package piece with brand new equipment, and after a month or two, they're all the same as the colonies started with old equipment or colonies overwintered. So no, we haven't published that yet. So again, that reminds me. And Megan has a paper that we need to publish too.
Yes, I know. There's that pile of unpublished data. I did recently read a paper that talked about that it might still be useful to do the treatments in cases where you're overwintering in sheds and the bees are really, really locked down for long periods. But like Zachary said, we just don't really use the treatments anymore after they kind of became unavailable. Dan, there's a question. It says, when making splits, is there one method that's better than the others for getting good honey production? And is there a better method for splitting when your goal is to increase?
Um, I don't think there's a better method. I think for if you're after better honey crop production, I think it's really it's about timing and size. Um, you know, earlier is going to be better just to, get more queens laying sooner. They produce eggs, which turn into bees, which are what's going to get your honey. So I generally try to make the splits about as early as I'm comfortable with, um, weather-wise and bee development-wise. I think the investment in purchasing a mated queen pays for itself in return in honey versus alternatives of dropping a queen cell in that you raise yourself, or, you're a couple weeks behind a mated queen, or, um, a walkaway split where you're kind of a month with nobody laying there. So I think investing in the mated queens for your splits is worthwhile. Um, and I think making your splits big enough, um, that if you leave the parent big enough, you want to trim it back to kind of relieve some swarming pressure, or but making that split big enough, and for me that's usually in the a full nuke box, a full 5 frame nuke, or if I'm making splits right into singles, 6 or 7 frames, because it's I find there to kind of be this little step at somewhere around 3 to 4 frames of bees, early in the season anyways, where temperature is certainly still an issue, cold nights. And smaller than that, they just don't quite take off. But once they get past that 5, 6 frames of bees, then they just, they grow rapidly. Um, so I think making them big enough and early enough, and getting as many, getting laying queens going right away, you're going to end up with bigger colonies sooner. That's going to capture nectar. If increases are your goal, I think, you know, you could still split early, but in Michigan we have a long enough season. You can make multiple rounds of splits if you want to look at, okay, my beekeeping season starts in, you know, April. I'm going to be starting splitting early May. You can still be making splits, as Megan said, into the end of July. You can get, a couple generations of splits, so you can turn, one into two, and that two might turn into five or six, and that five or six might turn into 10, if you get three splitting windows, and often you can go more than one to one. Um, and if you want to do it, if you want to do it on the cheap, maybe you do two cycles of splits, but do it with queen cells, or something like that. Um, and I'm generally, bees and time and food makes more bees. I find usually the investment in feeding splits, if I'm trying to grow numbers, I find that to be worthwhile, especially if you're limited on drawn comb. That's going to be something that accelerates your bees growth rate, both for either you've got supers, that's going to increase your honey crop, or just making splits and you've got drawn brood comb to give them that accelerates, or it doesn't limit colonies growth rate. So I think those are things where looking at giving the bees all the resources and all the favor you can, and then just really looking at the timing of things.
That was great. I could see Anna and I were just both nodding in unison to all the things you were saying. So, Anna, there's a question that says, I've had several colonies die of isolation starvation. How can I prevent that next winter?
Yeah, so I saw that in Minnesota in the winter of like 2018 to 2019. And then we saw it a little bit in RBs this year, but I hadn't seen it since then. So I think what they're asking about is when you have a colony and they're eating they're consuming the honey, and if it's cold for an extended period of time, the cluster won't break cluster to move to more food. So it will eat all of the honey that it's touching, and if it doesn't warm up, that colony can just starve in place, and you might find it's just like an inch or two away from stored honey that it just couldn't get to because it wasn't warm enough. So I think a lot of it, unfortunately, is just going to be bad luck. However, I also think that we can really try to organize the frames of honey in a way where there's not gaps, right? So that's why, especially when we're doing our fall management and trying to configure the hive, we're making sure that the frames of honey are kind of close together. If I have any frames that are partly drawn and still foundation, I try to move those to the bottom or the outside. If there's empty comb, ideally we're feeding heavy syrup so that the bees are filling that syrup into the empty comb in that brood nest area. So trying to make the food kind of as close together as we can in the top box and just not leaving a lot of gaps for bees. But some of it I think does come down to luck too.
All right, we're slowing down on questions. So if you do have some, you can add them in. Otherwise, we can always end after just a mere hour of questions. So this one, as Zachary, do you want to talk about, so the Noroa, the miticide is based on RNAi technology. I don't know how familiar you are with that product, but I know you have been working on RNAi things. Um, do you want to talk about RNAi and Varroa at all? And you're muted again. You're muted.
Yeah, yeah, it's difficult. It's difficult a little bit. It's very technical without the slides, but basically RNAi is a native mechanism within the cells that the most organisms have a method for, I think it's to fight against the viruses. So they have this RNAi mechanism that if you have a stretch of double-stranded RNA coming to your system, then that's going to trigger something similar to an immune reaction. Think of it as. So then they basically recognize that double-stranded RNA as foreign, and then they have machinery to bind it and open the double-strand RNA, and then so it becomes two single-stranded, and then that's going to match any RNAs that are complementary to those two, and then basically chop those up. So that's the mechanism. So the Green Knight's new uh, raw treatment overall, or with this
Naroa, yeah.
Loroa, or another names. I don't know why they have two names. Other names with the scanner or something like that. We, I don't know how it's pronounced. So in that one, they target calcium channel in the mite, and that apparently doesn't kill the mites, but reduce reproduction, so they have less babies.
Mhm.
So, but I'm not sure why they pick a gene. I mean, you would think they would pick a gene that kills them. All right, it would be faster. Uh, but I assume the drug will last, because that drug lasts like 6 weeks, they claim. But if you're killing the mice during that 6 week period, wouldn't that be better? I don't know. Maybe they're afraid of resistance or something if you're killing them too much. So that's basically, yeah, my lab, we did a study. We did about 6 or 7 genes, and we found we basically just stole the genes from ticks, because some cattle ticks, uh, genes that have been used before for RNAi. So we figure the mites are more related to ticks than to insects. So we use those genes to test, and then we found 2 genes that would kill the mites right away, and 3 genes would affect the reproduction, very similar to the commodin gene they used, the green light used. Yeah, so there are other genes you could target.
Mhm.
So so we were joking today with Jeff, saying we could introduce that gene to some microbiome gut bacteria in the bees. The technology is there, and then instead of using the green light gene, which is patented, you can use my genes. I didn't patent those genes. But of course, then we don't know how big a hurdle it is to jump over EPA and FDA, because the Green Knight did, but because it's artificial produced double-stranded RNA, it's going to leave that 6 week period in the sugar, then it's gone. But if we're going to genetic engineer into the bacteria, which is different, the bees are going to carry that bacteria, and we don't know whether honeybees will transmit it to bumblebees, to another bee. Does it come back? Does it go to another organism? But as long, of course, safety issues, as long as the gene you're targeting doesn't have the same similarity in bees, in honeybees, in humans, in other wildlife, then you're fine. So you have to make sure that raw mite gene is very specific that you're targeting, so you're not going to kill something else. You're
Mhm.
not going to kill some beneficial mites, if there's such a thing.
Yeah, it's funny to think about beneficial mites, but
Uh,
and I will say for the Naroa, the goal is to put it in like the bees that have to eat it are the young ones because that's the ones that the Varroa are going to feed on. So, we're feeding it to the bees 18 days after the split, so it kind of goes in that first round of the young bees. But I think Zachary's point about having a treatment that uses this technology is very new for us, and it does open a lot of doors, hopefully. Um, so there is a let's see, there's some good questions came in. Anna, there is a new beekeeper who's getting a package on April 10th. Is it warm enough to put them in the hive right away, even if they don't have drawn comb?
Yeah, so I found the links to a couple of videos that I'm putting in the chat now. So one is from Michigan State University on hiving a package, and then the other one is from University of Minnesota, and it's specifically on hiving a package in cold weather. But I would say for most situations, you want to hive them sooner rather than later. You don't want to wait because that's stressful for the colony. So like if there's like a tornado or something, you can wait. But if it's just cold or normally what we want to do is get them in the hive And then what we want to do is feed them sugar syrup directly above their cluster. So you want, in this case, you usually want a gravity feeder or a bucket feeder that you put directly above those bees, and you just want to make sure that it's not leaking onto those bees. And the bees will consume the feed and they will start drawing the comb. But I would say get them into the hive sooner rather than later, but really stay up, keep up with the feeding and the sugar syrup. Check them after you've hived them to make sure that the cluster is underneath that feeder and that the feeder is not leaking.
And I'd say the other thing, if you wanted to get fancy, Better Bee does have pre-made drawn comb you could purchase. So I put the link in the chat. I will say in full disclosure, I've never used it, but this is kind of like the target example of when people would use it.
So
There's
when I started MSU 28 years ago, I was using the Illinois calendar. So April 1st, I bought my packages and installed it. And then it snowed for 2 weeks. But the bees were fine as long as they have food and inside the hive it's warm enough. The cluster will keep it warm, especially if you bought 3 pound packages, and you'll probably be safer than 2 pound packages. Like if it's today, same weather as today for 2 weeks, 2 pounds might have a hard time for the queen to lay eggs and to slowly build up. But 3 pounds will definitely help. Yeah. So, and my bees didn't die. It's just I just felt stupid. I installed them on April 1st, but I think after that I usually do it April 10th ish, 15th. Yeah.
There's a question that I absolutely don't know the answer to, and I think the correct answer is going to be it depends, but I'm going to put it to the crowd to see if anyone else knows how long a pint of sugar syrup would remain in the brood area. My official answer is I have no idea. Yeah, and I think it really does depend on whether or not there's other nectar coming in and how quick they are. So but I also like that there's lots of questions that come in that we have no idea. There's another one that I think we're going to give an equally unsatisfying response to, which is why you're here. And it says, can you talk about the advantages of using insulated hives in the summer and winter versus wooden hives? So I have only used the polystyrene nukes. We do have a variety of interesting hive types for us to set up at MSU, including some insulated ones. But I don't have a lot of experience personally using insulated hives. I can say, though, that this is something that has been well studied because obviously we don't need new technology to try insulating hives. And there are some really interesting old publications, both from the Wisconsin lab. I know there's some from Wyoming and some from I don't know, there's the thermology of the wintered hive that also answers this question. I can't remember which lab did that. That came out in like 71. And it is something that people have looked at for quite a bit. And there's new research as well. It's kind of one of those perennial questions for beekeepers is how much to insulate. There's definitely not a huge survival difference or any survival difference in an insulated hive versus non-insulated in Michigan. Some of the older papers will talk about how they'll maybe start raising brood a little earlier in insulated hives, but it slows down once it warms up. So there are some differences that you see in the springtime. But there isn't anything in terms of like basic survival of honeybee colonies versus insulation. I don't know if Dan, you want to talk or Anna about more about the nuances of insulated hives.
I don't have a lot of experience. As you say, we have one we're gonna populate this year on campus, but I don't have experience with them.
But that is a fun topic to look at the old literature because, there's so many things that have changed, like we now have molecular testing, but, insulation is one of those where you can go back, literally people have been asking it for 100 years. Anna, there's a question about voluntary registration for cottage food operations. that potentially they're going to allow them to put their registration number on the labels in place of the address. And will this be available to honey producers as well?
So some of the MSU Extension educators we work with know that this is of interest to many beekeepers to be able to have, for example, MSU Extension address on their label instead of a personal home address. And then if there were any concerns from the Department of Agriculture, they would be able to get in contact with the beekeeper through that connection. So that's been passed along, but there's nothing official as far as I'm aware. We don't have that as an option yet, but people know that beekeepers are especially interested in having that option.
All right. And I like that I think we can end on this question. And it says, what are your Varroa treatment plans for this year? And so maybe I'll do mine and then Dan and then Anna, you can go, and Zachary if you wanted to add in. So I do most of my splitting with queen cells. I don't use mated queens, and that's just because I'm cheap and I like my bees. And so, but I agree with Dan, if you want more honey sooner, you would use mated queens. But for me, the other reason is that I will have a break in the brood cycle. And so that's kind of one of my main ways of the Varroa management is to do like a break in the brood cycle during splits. And then I am very interested in using Viroxan and Formic at the same time when I put my honey supers on. And then I've historically relied very heavily on Formic Pro and have done multiple treatments with that. There was a comment that someone put that they had problems with queen issues with that. That has not been my experience, and I've been using Formic. I used it when it was the Mite Away 2, the huge pads. I've used the Max. I've used the Formic Pro. We did a trial last summer in 240 colonies using Formic, and we just didn't see the queen loss. However, I think when a lot of people do see the queen losses, it's because they're not using young queens, and the queens tend to die. If you're not replacing the queens, then the queens are going to get replaced in the summer, and that's often the time that you're putting Formic Pro on. So it is useful to just pay attention to how old your queens are when you're putting it on. So I am going to do, splits where they are getting a break in the brood cycle. And then my next thing will be Formic Pro and Viroxan at the same time when you're putting the honey supers on, and then likely another Formic Pro after the 56 days of the Viroxan is up. And then I'm going to assess, and it'll kind of depend on where they're at, depending how hard I have to go in the fall. I know, Dan, do you want to, or Anna, you want to go ahead?
So I'm often thinking about it just like in terms of one management strategy per season. So spring, summer, fall, winter. So we have overwintered colonies that are coming through. So we're thinking about Formic Pro for spring, summer, and fall. And now that we have Viroxan, maybe swapping out one of the spring or summer treatments for Viroxan. But with our colonies that are big and established, I think that's good for Formic Pro. I think if you're starting with smaller colonies this spring, like a nuke or a package, Formic Pro is not going to be the right treatment because the colonies need to be at least a deep worth of bees. So then you're thinking about another treatment like Roxanne or maybe HopGuard. And then one thing that I'm really excited, so normally in the winter, we do an oxalic acid dribble or the solution method where when we expect there to be no or little sealed brood in the hive in November or December, we quickly dribble oxalic acid onto the bees with the mixed with sugar syrup. But there's a new product called AP Bioaxyl RTU for ready to use that's you don't have to mix up, and I think it is at a higher concentration rate and the bees don't ingest it. So I'm really excited to try that this winter. Dan.
Yeah, very much similar to what you've already said, but Formic Pro 2 times depending on where we're at in the fall. The Viroxan in the summer as kind of that long term. And it's nice to have multiple options like that that can be used with supers on. I, like Megan, have not had major queen issues with using formic. And we said that pretty careful about watching the temperatures and at least the first 3, 4 days not exceeding low 80s. Which in Michigan typically we are able to find that. I was really pleased last year that we did have an extended warm stretch, um, and also based on work schedule and when I was available to get out and treat bees, um, I couldn't find that formic temperature window, so I was really happy to have the Viroxan available. Because I kept watching weeks go by when I wasn't able to get formic on, so being able to get the Viroxan on, and I think working that in combination, those two products, and then typically at end of season kind of cleanup treatment with the um, oxalic dribble, like Anna mentioned. There's now the pre-made, ready to use stuff. And I did purchase a little bit of the Naroa just as a new product. I want to kind of learn about that, get some firsthand experience with it this season. As with anything new you're kind of working into your beekeeping, I would say don't, it's okay to kind of dip a toe in and get some experience before you commit the whole operation to something that is new and you're not familiar with. And like anything, there's going to be some nuances. And so I'm a big fan of trying something out in kind of a smaller scale before I figure out how to best kind of adopt it and put it in my toolbox for the pros and cons and learn what situations I want to use it in.
I also want to say that one of our best resources for the different Varroa treatment options is the Honeybee Health Coalition's Tools for Varroa Management Guide. And we are lucky as beekeepers that there are several new options for us. So the current guide that's out as of today, April 6th, when we're recording, is the 8th edition, which is missing a lot of the newer treatments. I'm on the committee that has been updating the guide. It's gone through a major rewrite. I think it's gotten a lot better, and it includes all the newer treatments. That's the 9th edition that should be coming out soon. So keep your eye out for it, and we will share it with you all when it's released.
And there are two follow-up questions to the Varroa. So one says, will you still harvest and use honey if you use Formic Pro? Formic Pro and oxalic acid, any of the acid-based ones you're able to use when the honey supers are on. So they are safe and labeled to use when honey supers are on. So yes, those are the ones you don't have to worry about removing the honey supers. There is a question of, do you use Apiguard in the fall? I often do use Apiguard in the fall. It does have temperature windows, and you do have to use that is one because it's oil based, it's thyme oil. You do have to take the honey supers are off first, but that is often the one that I use after I take the honey supers. And then there's a question about the Apibioxyl RTU if it's made with the glycerin instead of sugar syrup.
So I don't think it might be a trade secret. So I'm not. Ask us again next month, and we'll see what we're able to say.
Yes. And I've not used the RTU yet, but I'm as Dan mentioned, we do try to make sure we use everything ourselves. So this is my first role at using the Naroa and Apivar 2.0 this spring, and then hopefully the RTU as well. So those were awesome questions. Thank you everyone for coming and listening and sticking around, and great questions. And we look forward to talking with you next month.
Thanks, everyone. Have a good night.